Hi,
Hi, Jackie.
how are you?
Good. Thanks for agreeing to meet.
Yeah.
I I I first heard about you through Nanome, through Daniel from
Oh,
Nanome.
yeah.
I was talking to him and he said, "Oh, I actually know somebody else kind of in that space." Well, he said he the company. He didn't say your name specifically.
Yeah.
But then I looked up the company and I saw you were behind it along with Chris Witowski. Is that
Yep.
right?
hmm
Interesting. So
Yeah. We both are the co-founders of the company, so we both have a organic chemistry and natural product chemistry backgrounds. So I think when we first talked to Daniel and the Nanome group, it was maybe somewhat refreshing for them to, you know, be talking to scientists
oh
when we were
yeah,
trying to work through things and talk about stuff. So yeah, they're a great group.
Yeah, totally. Did they reach out to you or did you reach out to them? Kind of curious how you came into contact with Nanome, do you know?
So we were at the same conference together. It was I'm gonna forget the name, but I of course somehow I'm remembering the acronym right now, but not the name. I think it's called like PDDP.
Okay.
Was the conference back in March, and Daniel gave a presentation, and I was on a panel actually, and Chris may have given a presentation, but either way, we ended up meeting through that, and he we were really intrigued just by obviously the fact that when you can actually work with these things and manipulate them and really see them yourself, it makes a huge difference. And I think even we were shocked at how much of a difference it made. We've been looking at this stuff for years. Like it was, you kind of come at it with a little bit of a jaded perspective of, oh, this isn't going to be that great. It's just going to be some other thing. But then once you were in it and you have, the VR headset on and you're touching things and doing things, you're like a little kid again. So it was really neat.
Yeah, that's shattering. Yeah, so I kind of looked you up and you're an organic chemist in natural products and I guess your group discovered a terpene.
Yeah, so that was turned out somehow to be most of my PhD was finding weird terpenes from weird places, which was funny because then I ended up working in the cannabis industry. So then it was just more terpenes and more other things. So they're kind of everywhere. But the main one I think we got attention for was Darwinolide, which
Yeah.
is the MRSA biofilm inhibitor, and that one was from an Antarctic sponge. So Chris and I actually both worked on that project, so we both ended up getting a publication out of that one. So that was fun. But so we actually did our PhD together years ago, and then kind of went separate ways, and then ended up coming back um, through the cannabis industry, and then now our own company. So
You're from Florida. Where'd you go for PhD? Where did you
uh,
venture off
the
to?
University of South Florida
Oh, okay. Is
in
that
Tampa.
Tampa? Okay, that's what I
hmm
figured. Um,
Yeah.
and what year did you guys finish what year did you finish?
So I finished in 2016,
Okay.
um, and then he finished in 2015. So he was a year before me.
And then so in between finishing PhD and starting Psilera Bioscience, you worked for some sort of cannabis company?
Yeah. So, well, actually, I went and did a postdoc in Vancouver at Simon
Nice.
Fraser University.
Yeah, yeah.
Yeah. So I did a postdoc there for antibiotics drug discovery. And that was really where I learned how to sort of clean up and build libraries and like chemical libraries and do kind of a lot of the different just databasing and data scraping and all the fun that that entails. But most of our focus was on like mass spec and looking at trying to find new antibiotics from different bacterium sources.
Interesting.
But then around that time, my father was diagnosed with a rare form of dementia years ago, and there was sort of a bunch of things happening. I have some family kind of struggling with addiction at the time. You have sort of the opioid, epidemic happening, and Vancouver was a big hotspot for a lot of that.
Mm,
And so it was just between all those things, I said, okay, I've always been in kind of infectious disease, tropical, and like neglected diseases. And I felt like, okay, well, the neuroscience realm is just as neglected as a lot of these other, diseases I've been studying for a long time, or actually not really studying them as much as doing the drug discovery for them. And so I moved back to Florida and worked for as a formulation chemist for a while for a pharma company, just trying to learn the pharmaceutical side since I had always been pretty academic. And then it was around then that Chris was helping to start up a cannabis company here, and I figured that was a nice sort of bridge between, okay, I'm already a natural products chemist, but I wanted to learn pharma, and now maybe I can help sort of bridge those worlds a little bit with the cannabis industry and really start doing some of the neurological healing things that I thought were really important. So yeah, we helped build that company for a few years, and then um, he was like the fifth employee,
Wow.
I think, and now they're over 700. The company is
Whoa.
one of the biggest private cannabis companies in the country now. So
Wow.
yeah, so we then when we kind of saw the psychedelics thing happening, we said, okay, well, that has so much more data behind it, and it just had so much more of a drug discovery aspect to it already, and there was so much information, and there's still so much we don't know about cannabinoids and cannabis. And after being in that area of research for a while, I'm still a little bit dubious that we're going to find something better than the plant itself at this point, because it's
Yeah.
just been really tough to really try to tease out individual molecules, which is what the FDA really wants when they're talking about pharmaceuticals a lot of the time, because they're still not sure how to deal with complex mixtures sometimes. So when we were looking at the psychedelics world, there were definitely compounds that had a little bit more of that traditional pharma feel to them. And not that that's necessarily the best route, but it's the one that we can work with right now. And so that was why we started Psilera, to try and, yeah, see if we could help build some new drug discovery sort of platforms and things with those.
Yeah, I think it's interesting you're talking about these complex mixtures, and especially with cannabinoids, you have like these more racemic mixtures, where you have problems with separation. As you know, natural products degrade really quick. They're
Yep.
not very stable, and it's really hard to separate enantiomers. But with
Yep.
psychedelics, the tryptamine backbone doesn't have any enantiomers, and that's really nice in a way. Like
Right.
serotonin and psilocybin, those don't have enantiomers. So that could be easier in that way to work with.
Definitely. And they're much smaller than LSD to when you're trying to synthesize these things, because LSD does have an enantiomer, and there's a dramatic difference between the activity of the plus and
Correct.
minus enantiomer of LSD. So it's definitely something you have to keep in mind. As soon as you introduce stereochemistry, it makes everything more complicated. So definitely, being in the natural products world, we're used to having really funky, unstable molecules with lots of stereo centers. So trying to avoid that as much as possible, I think, is, if you can. I think we're in sort of a unique position where these compounds weren't really allowed to be worked on in certain ways for
Mm,
a long time.
yep.
Otherwise, this would be totally low hanging fruit decades ago. Like this, it's just a rare position to kind of even have the opportunity to start being the one doing this discovery. Whereas, any other simple molecules like this from nature would have already been worked on decades ago at this point. So I think that it's a really nice place to be. And now with all the technology we have, it's even more unique, and we can do it really, really well and have a good foundation now, rather than sort of playing the guessing game that would have happened a long time ago. Because even the serotonin receptors, I mean, so many of those have been broken into subtypes just in the last 20, 30 years. So, I mean, when
Mm.
they were doing original assays and research, back in the 60s and 70s, even the 80s, sometimes you'll see data and they still haven't even separated those receptors into subtypes yet. So it's hard to even really know what activity they were looking at sometimes.
And yeah, you sparked a thought in my brain. And I guess the thought was that like you're talking about serotonin subtypes, and like SSRIs are like pretty, as of now, I'd say they're pretty crappy drugs.
Yeah.
They don't have they're just like selective serotonin reuptake inhibitors. Like what subtype? It's just like, all, it's fine. Do it all, no
it,
problem.
it just hits all of them.
It's all of them. When in reality, we're trying to sort of, with the psychedelics, we're running that, no, there's selectivity towards 2A with psychedelics, and maybe less with 2B and 2C, but we still don't know the physiological functions of a lot of these receptors on a very specific level.
Yeah. Yeah. And I think that we at least were able to do a lot of the experimental side quite a bit with these, which obviously there's a bit of an argument there, right? So just, full disclosure, my spouse at home, he's actually a biomolecular physicist. And so his
Oh,
focus is on protein dynamics and how they
that's
move
awesome.
and
I like this stuff too.
salvation and stuff. And so when I try
Yeah.
to tell him, well, no, they tweaked this part of the molecule and it changed this in the activity, he's like, I don't care.
No.
He's like,
Yeah. No.
he's like, that's not how it works.
He's like the
And so it took
deeper level of computational chemistry. That stuff's great. I love it though.
Yeah. So I think that there are definitely some instances where some of that experimental data can be very helpful. And then I think there are other areas where you have to remember, okay, we still can't fully explain what's happening in the receptor and what's really going on there. We're not even really close sometimes, unfortunately. So I think it's just good to keep that stuff in mind. Know that this is kind of the best that we have right now, and we should do what we can with it. But always remembering its limitations and to know that it can still be better. So that's sort of the realm I try to stay in mentally when I'm doing this stuff. But I think that especially just in the last couple of years with some of the different crystal structures, obviously the Roth group and others with and
Yeah,
I've seen you do quite a few, of your videos on the LSD and 5-HT2A sort of scaffolds. But I mean, even there, when you really read the publications, they're kind of reaching a little bit when they talk about, why maybe it's got the longer duration or why this and sort of what the interactions are. And all we can do is the best we can with what we're seeing and what we've got. But know that, hey, there could be something, later that we find that's out of left field and very unique. But so far, I think we're getting a good picture of what's going on. So I think
totally.
yeah.
I think I guess like my first frustration going into a well, not my first frustration, but I came from a chemistry background, right? I came from like
Yep.
organic chemistry and chemistry in general. And then I go to a pharmacology lab and I noticed a lot of the compounds were working with the racemic mixtures. And I was like, no. And then I asked my boss if you could have the chemist, like, is there a way, do you guys have a chiral column? Can you separate them? And he said, yeah, we actually can. He was able to separate them for me, which was like my dream come true,
Yeah.
because you know this more than anybody, right? Uh, receptor binding sites are stereoselective.
Mm-hmm.
One binding site can be selective towards an R enantiomer, one can be selective towards an S enantiomer. Just because the chemical formula is the same doesn't tell you anything about the binding at a certain receptor site. So I think that was my big realization when I first entered graduate school. It's like, wait, they're using racemic mixtures. What's going on here?
Yeah, and that's a big deal. I mean, enantiomers sometimes can even be a bigger deal than diastereomers. Like if you've got, 7 chiral centers and only one of them's different, that's not going to make nearly as big of a deal as when you're dealing with the complete mirror image. Like things are going in the opposite direction. Like that's what that means. So, and we've seen it. I mean, the other big one, obviously LSD has some different activity, but one of the ones I used to always use sort of as an example was gossipol,
I
which
don't
is
know this one. What is
it's the
this?
toxin from cotton usually.
Oh,
And this was discovered, decades ago. So in my mind, I'm like, we know this exists. Like we know this is a problem, but the R and the S enantiomer, one is much more toxic than the other. And they first were kind of looking at it with, I believe, rabbits, like kind of eating cotton and that sort of thing. But then they started to learn how to genetically modify the cotton to remove even one over the other as kind of like a preference, just because one was a lot more toxic than the other. And I think that one, I can't remember when that was done, but that was a long time ago. And I mean, that was just kind of an example of, well, this one's toxic and this one's significantly less toxic to the point that we're not even really worried about it. So, and it's a very small molecule too. It's not a very large compound. So it can make a big difference.
yeah, totally. It reminds me of thalidomide, right? That's a big
Yes.
one. That was a
Yep.
disaster,
It
unfortunately.
was.
That was a disaster. That was the
Yes,
worst one of them all.
it
But
was.
yeah. I mean, stereoselectivity is important and being able to separate. Do you use a chiral column for that? Is that the way to do it? Like, how do you do
Yes,
that?
yeah. Typically, if you and chiral columns are obviously
They're expensive, right?
expensive. They
Yeah.
are. I mean, I think we've had some chiral columns that were 5 times the price of just like the equivalent in a C18 column. And C18 columns can already be pretty expensive. And so they can be pretty outrageous sometimes. The Obviously, if you've got something that you can volatilize, There are some more unique kind of ways, because obviously GCMS is pretty powerful sometimes with separation. And so this is not perfected by any means, but we even had a GC fraction collector at one point trying
Wow.
to, collect the different fractions of different volatile compounds. And that sometimes can be a little bit easier with separating out really, really similar compounds. But the fraction collectors they still need some work. They're not ideal for collecting compounds, unfortunately. And then I think at least in like some realms, if you've got something that can be distilled, sometimes distillation can still be a pretty good method, which is funny because it sounds almost like archaic, like alchemy at this point,
Yeah,
but
yeah.
but actually it can still be, because it's a similar concept kind of, GC is still volatilizing things. And but yeah, otherwise if you're doing liquid chromatography, it's usually chiral columns.
Good to know. Good to know.
Yeah.
What was I gonna say? So I guess like in the sort of psychedelic space, there's all these papers that come out, right? And one of them was the I guess the bigger paper that came out was that microdosing is basically the same as placebo. You probably saw this, I'm sure.
Yes. Yeah.
And it's not to say that like microdosing definitely doesn't work. And it's like I definitely trust the Carter Harris group and I trust their research. So I'm sure they did a good job, but I am curious to see those scale up and see what happens in the world of microdosing to see if it's actually beneficial.
Yeah, I think that's sort of the question on everybody's mind. We actually did a blog post on this a couple weeks ago on our website, just because I was even getting kind of questioned and asked. And obviously, we have investors and family and people around all the time who are kind of keeping an eye out for us on stuff and saying, well, hey, what do you, because that's really one of our target models is more on the microdosing side, but specifically with DMT. And rather than psilocybin or LSD or some of the other options. And that's really because there was data a few years back when they compared sort of DMT, MDMA, ketamine, LSD, psilocybin, a few DOI. I think there were others,
Yeah.
but DMT was the only one that still showed efficacy at subpsychedelic dosing. And it
Interesting.
was for kind of neurogenesis, neuronal growth, neuroplasticity, that sort of thing. And so we're really uh, Chris's family has some Alzheimer's in his background, and then my father has dementia, and I've had dementia in my family. So neurocognitive disorders is a big focus for us. So if this is still showing some kind of neuronal growth or plasticity at subpsychedelic dosing, then that's a good microdosing type target. Which really obviously microdose is just low dose. I just thought it was
Yeah.
hilarious that that became a thing. I'm like, isn't it just dosing? Like what?
Yeah.
But I mean, yeah, I absolutely also trust the Carhart group. I mean, they're obviously ahead of the game with a lot of these studies. But I had the advantage at this is a different conference back in November, I want to say maybe, for it was a psychedelics conference with Hanson Wade, and they
Mm.
actually, I'm going to butcher his name, apologizing in advance, but he's on the papers a lot of times with them, and it's David Zitsoy. It starts with a Z, I believe, and he's usually on the
Mm.
publications, and he works with Robin's group. Um, he presented on that data before that paper came out.
Mm.
And I asked him, well, what kind of control did you have over the dosing for these people? Because you're crowdsourcing, right?
Yeah.
And he's like,
Posting on Reddit
I
and
was like,
stuff. I
I know
saw
you're not a
these.
drug dealer, so I know you're not just handing this out to people. And I know that the government's not letting you do that. So, how are you really controlling that? And he said, well, we're not. We have to just trust them. And I was like, so that also means you have to trust whoever is giving them that product, that they actually know the potency, which they don't. And microdosing has no definition at all then in that case for
Mm.
whatever they were taking, what they were given. And it's a tough study to do because we are in a weird, hairy kind of regulatory zone, right? So I both very much appreciate that they took that risk and they did all of that data and they put they showed something at least out of it, but I don't think anyone can draw any conclusions from it just because there's no control over any of the doses or the data. And I mean, I'm not, again, most data is showing that, at least on a cellular level and receptor level, some of the microdosing options don't seem to be as efficacious, specifically with psilocybin. But again, that's at least one of the reasons we're really looking at DMT and trying to make sure that the compounds we're looking at are ones that have been shown to maybe, do something there. But I don't know. I still think that there's a lot of benefit to it. I think that there's too many people around the world who've been doing it. And even if it's just for sort of in between mood maintenance or whatever it might be, it seems to be doing something. But trying to tie that then to an FDA approved indication is going to be pretty tough, I think.
Yeah, no,
Yeah.
definitely. Yeah, I saw a lot of those. There are a lot of those surveys that go around Reddit and stuff like, oh, I'm a researcher and I'm looking for like people that have done this, this, and this, and do the surveys. And that's essentially
Yeah.
what the Carr Harris group did for it. I guess like from their perspective, like what else could they do? They can't, I guess they probably
Yeah.
couldn't legally have people in their lab like, oh, here's your microdose,
No,
Although
not
maybe
on
in the
that scale
future
at
that would be
least. Yeah.
great. That would be great to do it that
Yeah.
way.
And just having, and there was it's funny because after being in sort of the cannabis world, which is just its own universe in general, it's
Yeah.
just a crazy place, right? And so to be honest, and so being a researcher and scientist by training, I always kind of find myself in this weird kind of in between zone between lots of different kinds of people, which is both fun and terrifying at the same time. But one of the things was recently they did like a mushroom cup, kind of like they used to do the cannabis cup to like see the
Mm,
best cannabis. And
yeah.
it was someone out in California did it for sort of like a mushroom thing. And so obviously this is just entirely illegal. I'm not necessarily promoting any of this. They put it on there and they were talking about it. But the interesting part was that this person runs, or at least is friends with, I'm not sure how they are associated with a lab. So they're actually able to test all the potency. They were actually
Mm,
able to validate and look at all of the potencies of the various entries into this competition, right? The range of potency was anywhere from 0.3% all the way up to 3%. And so it's
whoa.
like when you have a range like that, that's what I mean when I ask you what your control is over dosing, is if that's the kind of stuff that's just being, sort of grown and handed out to the public, like that means that's too big of a jump. There's not even close to being like a small range of error. So I think that at least that just came out maybe a week or two ago. When I saw it,
Wow.
I thought to myself, this is
Wow.
it. This is what really shows you how variable the concentrations of psilocybin and psilocin can be when grown in different conditions, grown in different places and different media. And when you change the environment of an organism, you change the chemistry. And so it was a really, I thought it was really neat. It was kind of like validation for myself a little bit. I was like, hey, see, look at that range. That's way too big of a range.
Yeah,
But
it's like the ecology of looking at all that stuff, right?
yeah.
Collective ecology.
Hmm.
That's interesting. I actually never really considered this, to be honest. But yeah, it's
Oh, really?
fascinating. No, I never considered this even as a scientist. Like, oh, I didn't think about that. Yeah, you grow in a different climate, different soil, different temperature. It's going to affect the profile of the, I guess, cannabinoid or the type of psilocybin or psilocin derivative, I guess. It's interesting. I never thought
Yeah.
about this. Huh.
And it's because a lot of those compounds are usually meant for either attracting other organisms or defending against other organisms, or they usually have some kind of purpose in ecologically in their life cycle. And so when you're changing those things, their body's going to put energy into different places at different times to produce different compounds depending on what they're interacting with and what they have. And stress, that's a big one too.
Wow.
So it can vary wildly. So
Wow.
yeah, so I love that stuff. Obviously, it's very different from the computational side of things, but looking at even just some of the, yeah, it's too many variables. It's going from in the world of computational chemistry, right? You have your box basically. We have a nice
Yeah.
box. We like a lot of people working in that box like their box, and then as soon as you go outside of it, there's too many variables. Well, ecologists are the opposite of that. It's
Oh
like
man.
we're going out into the world where there's 387 variables and we have no idea what's going on. We're trying to make sense of it. So yeah, very different.
One of the, if stand me corrected, but I think 5-bromo DMT comes from like a sea sponge, doesn't it? You'd probably know about this.
Yes. So I think there's five, and then there's also six bromo. They're
Oh.
both of them, and they Man, yeah, I'm being a terrible marine chemical
No, it's
ecologist right
fine.
now, which I do have a background in it, but it was, I can't remember. I believe it was a sponge for that one. And the only reason I'm tripping up a little bit is because there's actually another molecule with a 2-bromo, which is
Oh,
very weird, very unique.
that is
And
very weird.
that one, and it wasn't DMT, it was just monomethyl.
Hmm.
Um, and that came from a coral. And I remember that one, but I think the 5 and 6 bromo came from a sponge. Bromination from marine organisms is pretty common though,
Interesting.
more so than terrestrial usually. And I actually, the very first marine natural product that was ever found was this highly brominated sort of crazy looking. It wasn't an indole. I think it had a couple other nitrogens going on, but it was, yeah, it was a wild little, tiny little compound, kind of like an indole though. But yeah.
Yeah, it's super interesting. So you guys, are you guys at Psilera like trying to, you talked about DMT and trying to make that, maybe for um, neurodegenerative disorders in a way.
Mhm.
Um, are you focused on like just the like are you focused in that way like just trying to create an established protocol for like DMT used at low doses for like neurogenesis? Or is it like you're looking at the scaffold of like, oh, I could put like a 5-fluoro here, I could put it, that kind of thing.
So we're doing both, actually, and that's because we both recognize the power of the natural compound itself, DMT.
Yeah.
And it's actually one of the most widely spread psychedelics out there. I mean, I think it's like in 50 different plant species. They even claim that you can find it endogenously in humans and other animals. So we recognize that has a lot of power as a natural compound. So we both are working on different delivery techniques and
Oh,
formulations of DMT to help with the neurocognitive, neurodegenerative, but even mood kind of. A lot of these neurodegenerative diseases come along with a lot of mood swings and other mood issues too. Obviously, I think if I was losing my memory or losing sort of my cognition, I would be freaking out too. So that's part of it too, is just looking at some of the symptoms of those diseases and seeing if it can even just help there. And that's one of our big focuses. And we're actually hoping to start a phase 1 clinical trial for
oh,
that by the
congrats.
end
That's
of this
great.
year. Yeah, fingers crossed. But that's one avenue. The other side being, the chemists that we are, it is definitely looking at, how we can sort of modify these scaffolds. We're not we're not into doing huge major modifications, and that's again because we recognize sort of how good a lot of these compounds are themselves, and the smallest changes to these compounds can dramatically change their activity. So
Yes.
we're trying, yeah, we're trying to find that sweet spot a little bit. One of our investors compared it to dancing through the raindrops, which I love that like thought in my head of like trying to find the best sort of spots to really
Oh,
fit
I
in
like that.
with,
Yeah.
yeah, with what's already been sort of done and what's new. And we so we're working off the DMT and the psilocybin scaffold to try to do some different modifications on those. That's the stuff that we actually, so you had mentioned in our video the image of DMT in the
You
5-HT2A
saw
receptor.
this? That's funny.
Yeah, and I did see you asked about that, and so we actually ran a lot of our compound library, our proposed compound library, through all of the currently known serotonin crystal structures, as well as sigma 1. And so we actually did some docking, and that was with our collaboration at the University of South Florida.
Oh,
But that also happens to be my spouse
is
because
that's not through Scripps, is it? Who's that through?
no. So
Let's
this
see.
is just the lab that, so his name's Dr. Woodcock. He's at USF, his lab, but um, so Stephen Austin is actually the postdoc that works with him, and he ran our compounds as well as known psychedelics through all of these different receptors so that we could compare known experimental activity to what we were seeing computationally as well, just to try to sort of validate what we were really seeing. And then to try to, once we kind of validate, okay, they seem to be tracking pretty well, then we feel better about some of the results for some of our new compounds, since those aren't known. And we've done that with about 60 compounds so far.
Wow.
Yeah.
Wow.
And about 40 of those are new chemical entities.
Cool.
The other 20 are known psychedelics or known sort of scaffolds that we've added to the mix. And we're using that to really help then guide what synthesis we're going to do. And so we've been synthesizing new compounds for about a year now. We have 5 or 6 now that we've synthesized based on, kind of what looks promising computationally. And then in the next 2 or 3 months, we're going to start animal studies with some of those compounds. So,
very cool. Maybe the
yeah.
HTR, those types of things,
So yeah, this one is actually a specific to alcohol addiction
Oh,
and
okay.
alcohol consumption. So we are looking at some substance abuse and like addiction kind of things. But she does also have mood, let me see, different behavioral assays that will just help show which receptor it's kind of hitting. So the main focus for us is 5-HT2A, so head
Of course.
twitch, those kind
Yeah.
of things. But she can actually do others as well. So that'll be kind of our first, sort of big test to kind of see, okay, is what we both predicted, just or hypothesized, I'll say, from as being, organic chemists and natural products chemists, to then what we've seen computationally, to then can we, sort of validate all of our minds and intellect and things that we hope work out right with the experimental data. So yeah, hopefully that all matches up. And then the last round will be, we do have a collaboration with the National Institute of Drug Abuse as well, and they're going to screen some of our compounds just against the actual receptors.
Okay.
But we were really big on trying to go into animals as quickly as possible, and that's only because there's still a lot of assays you'll test and you'll see activity in the enzymes, and then you put them into an actual organism and it just doesn't work out. So we are trying to do both in parallel rather than sort of starting with the enzyme assays and then jumping to animals and then having the potential of it not looking the same. We do ADME prediction stuff. We tried to do what we could computationally to help with how it might be metabolized, but I'm not, I don't know how much you know about it, but DMT is not orally bioavailable.
Yep.
So, if we're working off of that scaffold, then we have to be kind of, careful with making sure even our compounds are going to be bioavailable in the way that we think, or that it's not just getting eaten up by your metabolism. So all the animal studies are IP, like IV injection kind of stuff. So that shouldn't matter initially, but if we can, really target, I don't know, patient-friendly options. I'm not a big fan of the IV method if I can avoid it. So, but yeah.
Yeah, that's a little more invasive. You have to have somebody into your facility, have a nurse kind of administer, a phlebotomist or something, which is not as patient friendly. But that's the way I think ketamine injections are done this way, right? When you go to a ketamine clinic, they're like IV injected, I'm guessing.
Well, so they also have the nasal,
the S-ketamine nasal.
option. Yeah, the S-ketamine option, which that's the Johnson and Johnson or Janssen sort of, method, which was the nasal option. But which we are actually looking at some of those as well, just because it's a nice way to avoid having to do IP or IV, and getting it still to the brain the way you need it to. Yeah, ketamine's an interesting one though, because that's one where they tried to sort of make the argument that the S enantiomer was a lot more powerful than the R, but I think it turns out that it's like really not nearly as potent as they thought, and that using sort of the racemic mixture or whatever people are kind of getting off the streets is just as powerful. So I think it's been tough. It's been tough to keep that one as sort of like a private market, I'll say, because a lot of people are still recognizing that they're not seeing like a huge difference, I think, between the effects when you're doing the pure S enantiomer.
Right.
But that may not be true long term. I don't know. I just know that was some of the arguments that have been kind of heard floating around. But yeah.
Yeah, I'm curious to see where the psychedelic space goes, because right now we're in this very this research could have been done like 20 years ago, but now here we are in 2021 doing this research. And one of the things that, there's all these different things that come to mind. And some of the other ones that are interesting are there are other papers that come out that say you might not even need the psychedelic effects of psychedelics to have a long-lasting positive benefit. And there was a paper also recently put out that they use ketanserin. You probably saw this too. They use ketanserin to
Yes,
block
yeah.
the 2A receptor. Then they administer the psychedelic in mice and rats, and they see these positive, long-lasting outcomes for like anxiety. So it's like, do you even need the 2A receptor for that? I'm not sure. Right.
And that was one of our original targets from the very beginning, was trying to reduce the 2A activity while maintaining some of the other activity. And that was really just because, especially if you're trying to look for something for substance abuse, the more you can reduce that, the hallucinogenic effect can provide a little bit of a mental outlet that's almost too enticing.
Yeah,
And so, and
agreed.
that's when it becomes difficult for any kind of substance abuse or addiction. And even though, you know, psilocybin, they've done studies on it to show that its abuse potential is extremely low. Like the fact that it's a Schedule I drug doesn't meet any of the criteria really for that scheduling. They tried to get it down to a Schedule IV, I think. This was out of Johns Hopkins, like in 2016. And they were talking about the abuse potential was extremely low, you know, negligible. But I still think that there's a certain amount of just what I'll say is kind of like a personal enjoyment of that escape that people will have. And so even if it's, and I don't really know how you quantify that, but you know, there's that aspect of it. So our hope was if you can reduce some of that HT2A activity, then maybe you could still have some of the beneficial outcomes. So when that paper came out, actually, we were really excited about it because that was quite a validation behind a lot of what we've been trying to do. So we were excited about it. Yeah, I definitely think, especially on that substance abuse and addiction side, that's going to be important if you can limit some of that. But I think everybody's different, and there's no silver bullet, and there's no, you know, there's no like one size fits all. And so I think that that's really important to remember with psychedelics, is there's going to be ones that are helpful for some people and not helpful for others. And I think there are going to be situations like treatment resistant depression that you might need some very large dosing to really sort of kick someone into gear mentally to work on themselves and work through a lot of maybe their traumas or something else. And then there's going to be other indications where you don't need that. That's not necessary. And, you know, trying to get my veteran father into a room blindfolded with a stranger, like, forget about it. He's never going to do that. Never in a million years will he do that. So it's just finding all different options for different types of patients and making it as personalized as we can. And you know, obviously one of the big ones too is the cardiac issues sometimes. Like there's quite a few of the psychedelics
The
that really
2B
cause.
receptor, yeah.
Right. And so that's why we included all of those different receptors in our screening, was because we wanted to see, okay, what might be hitting 2B, what might be hitting 3, you know, can we help out with some of the nausea, you know, or maybe add some anti-anemic, I always mess up that word, emetic properties. um, and then, yeah, just the different receptors kind of do different things. So that was one of the reasons we made sure we included all of them. But it was that interplay, I think, is going to be really fun for figuring out sort of like why different psychedelics cause different outcomes and different sort of reactions to people. And I think it's going to come down to sort of a fun little like harmony of different receptors being, you know, hit in different ways. And yeah, I think that looking into that a lot is going to be interesting over the next few years. But
And maybe you can figure that out with, like, you're talking about this idea of personalized medicine is really what you're getting at. And it would be interesting if you could just, like, you know, somebody had a method for just like, oh, let me get a cheek swab, and then based on that cheek swab you could, you know, sequence someone's DNA really quickly and be like, all right, these psychedelics are good for your indication. That would be cool.
yeah,
I mean, we're probably pretty far away from that, but that's the dream.
yeah, like if you could find a way to have like biomarkers for people to know that right away, like, okay, it looks like your makeup of, you know, you have this many of this type of serotonin receptor or this much of this one, and you're gonna need this much of each type of thing. And yeah, because there's always there's so many variables. Because I always used to say, like, the human body, it's like so when I did chemical ecology, I was like, well, that's just drug discovery of like the human body, is just trying to
Yeah.
sort of mess with the ecosystem of our bodies. And uh, one thing that's also going to be really cool is looking at like microbiome stuff, because your gut has so many serotonin receptor and just serotonin in general is very prominent in your gut area. And so it's like any of these orally available options, I'm sure there's a pretty unique experience or outcome happening there rather than maybe even just transdermal or IV or something else, just because of how it's interacting sort of through your own sort of microbiome and stuff. So I don't know. It'll be fun to kind of see how this stuff plays out. That's why I like to say we're still in very sort of the fun drug discovery and development stage of this. Like there's a lot of people trying to jump right into like the phase 2 clinical trial realm and sort of just, that's like kind of just throwing the natural product sort of at the wall and hoping it sticks, and I think that it'll work for some. But I think for others, we can optimize it. And if we can do that at the beginning and create a solid foundation, then it's worth trying to do that now. Because psilocybin it's a great compound, but I don't know that it's ideal for trying to work with it and trying to put that into different medicines and actually have that be the active drug. I think you can make something that's going to be more stable, more shelf life, have different aspects to it that's going to be better than that one. But, and not to hate on natural products, obviously that's my background, but like you said at the beginning, we all know how stable they can be sometimes. So yeah, we'll see where that all goes.
Yeah, I'm really curious. Something that also sparked my interest or confusion in pharmacology is, you read a lot of papers and you see like, oh, this drug has this effect on receptor, and it's basically two dimensions. It's looking at a dose and a response. But
Mm-hmm.
in reality, drugs do much more than that. And I think you can probably use you guys have probably a bit of the in silico stuff, maybe machine learning, where you can say like, this drug has this affinity, it has this effect, it has this bias towards beta arrestin. You can sort of put together like 5 or 6 different variables for one drug profile. I think that would be cool.
Yeah. Yeah, I definitely think trying to collect as much information and as much like data as you can behind what's sort of happening along the way is going to be a key factor, especially to just any kind of success trying to bring a drug to market. I think it's just there's so many other variables. You got to be able to know what it's doing and what it's doing along the way, and sort of, yeah, what's it hitting, where's it going, what else is happening. And a lot of people will say, well, can it cross the blood brain barrier? And I'm like,
Exactly.
but how did it get there? What did it do before it got there? I was like, there's so much more that it can do as it's going along that path. And that's an important component for sure. But obviously there's a lot going on. So it's like, well, what else can we look into? And we know so much more about metabolism too, and other
Yeah.
things that it's like, it should really just be fully integrated at this point as part of the path. Like when you're doing this drug discovery, I feel like it's not supposed to just be sort of an afterthought anymore. But I think a lot of people still treat it that way, which is, um, but that might also be because it hasn't been around forever. I mean, I think I like to think it's been around forever, but I think it's just because maybe during like the time that I've been in school and doing things, it's been around. Whereas obviously, if someone's been in pharmaceuticals for 30 years, that hasn't always been an option. So I think yeah, I don't know. I think that stuff's gotten pretty good though at predicting those things. So I don't really think it comes down to saying, well, I don't know, it might still be wrong. I feel like a lot of that stuff's gotten pretty good at predicting, especially the really well-known metabolic pathways, like even your
Mm.
CYP, like any of the oxidation sort of enzymes and stuff. I would think it's pretty good at that now, but yeah.
Yeah, I guess you as anybody else, or you especially would know, like you have THC. That's not THCA is really what occurs in the plant, then it gets metabolized into THC, right? The acid form. And psilocybin, gets metabolized to psilocin, which
Yeah.
becomes orally bioactive. So I think, it's really hard to design a drug in that way because you have to account for all of these also pharmacokinetic variables for sure.
Yeah. Yeah. And I mean, I think that most people that were originally sort of jumping into the psychedelic space kind of went straight for the, okay, well, psilocybin actually turns into psilocin, like you said, and that's actually maybe what we can work towards. And so I know that there's a lot of groups doing sort of like pro drugs and that sort of thing to try to just have more stable options. But yeah, even then, sometimes you introduce any functional groups that could start targeting a different enzyme then, and then something else happens, and then you have a different metabolite coming out. And so, yeah, it's sort of a game you have to kind of play along the way. But yeah,
Totally.
yeah,
What was the like I do my boss does tell me a lot that scientists do have like they have a research group or whatever. And then they're like, we want to start a company. And so they spin off sort of a company. Is that like that seems so intense. You're just like, we're gonna start a company. Is that just eat up all your time? And like how intense is that
yeah,
trying to
it's
start a
pretty
company?
intense. I mean, again, thankfully, Chris and I both worked in a startup pretty much in cannabis. And so it's like we've already been part of that process and had to deal with sort of regulatory ambiguity and kind of already be part of a world that was a little bit academic and research and new, but also still somewhat established. And then you had to deal with manufacturing and production and a lot of other sort of variables. Um, Starting our own company, the thing that I think was the best was that now we can really make it what we want it to be, in the sense that we know we're great at research and R&D and early stage drug discovery. That's our entire backgrounds. And not to like pat ourselves on the back too much, but we're pretty good at it at this point. So if we know that that's really our strength, then it's not killing your, like, the way you're going to be successful is just to not kill yourself trying to be great at everything. So it's okay, this is what we know we're great at. Now we need to start finding people we know are great at the other things we need help with along the way, and create a team so that we can then help move this all forward. I mean, initially you have to wear a lot of hats. That's just sort of the game. Like it just happens, and you're gonna have to do a lot of administrative stuff and other things you don't
Mm,
want to deal with, and you just kind of put your head down and do that, and then get back to the fun stuff sometimes. But if you can really get through that first year or two of kind of just wearing all those hats, and it is a little bit stressful and dealing with the stuff you might not always want to be doing, and build that team so that different people are going to handle the different aspects that maybe you're not quite as good at, to get you back to where you are great, then it'll be successful. It's just, I think where a lot of people end up failing too is it's just a lot of work. It's a lot of work and a lot of time and a lot of effort. And it's a lot of, you never know who's going to end up being someone that can help you along the way too. So building up just your group of people that or just trying to get out there and meet as many people as you can, and really trying to bring in as many minds as you can along the way, is a huge aspect of it. I mean, I kind of lived my life like that from a young age, of like, well, I never know if I might need somebody someday, so I shouldn't ever be treating someone as if they're any less than I am. And I think that has really led to, us now having a huge sort of repertoire of people who are coming out of the woodwork saying, "Hey, like, I remember you. I'm good at this now. I've been doing this for like a
mm,
decade. Can I help?" Or this. And we've had so many people trying to just help us pro bono. Like, they don't want any money. They just want to help
wow.
because they love the idea of psychedelics and they're doing well for themselves. So they just want to kind of like help where they can, especially scientists. Like, because so many scientists just want to be part of something unique and interesting. And so the research side, yeah, we've got that covered for sure. But it's all the other little things that do
Mm,
become a bit much when you're starting a company. And but yeah, again, if you can get through that initial sort of stage and the initial, like, grunt work of it, and then just hire on people that can help you out, it always, I think it'll work out. I think it usually will for anybody. It's just, yeah, trying to get through that.
yeah, that sounds daunting to, well, it's not, I'm not planning on doing this myself, to be honest. It sounds, I like my box, I like my
Yeah,
little, I
see,
like my
exactly.
box. I like my
Yeah.
in silico box.
Yep.
But
Yeah.
I was talking to a fella at Cygen, which is there's all
Yes.
these startups, and
Hmm.
they're out of Calgary, and they're just focused on, making pharmaceutical grade psychedelics that somebody maybe running a study could buy from them, and doing
Yeah.
that through the Health Canada, I guess what it would be in Canada. But you're sort of taking the approach of like synthesis to
Yeah.
well, in silico to synthesis to in vivo testing and then to clinical trial. That's insane to me. How many people
Well,
work for the company right now?
so full-time employees right now, we actually only have 3. But again, we have a lot of academic collaborations and a lot of
Yeah.
other collaborations that have been huge assets for us. I mean, huge. And one of them is the computational work. The other is some of this mouse model work. And with NIDA, like those are both academic or government institutions. You know, like we've we've tried to, even though that might make your timeline a little bit longer when you're working with government entities, we know that their data is going to be really good. We know it's going to be robust, and we know that they're going to put their heart into it because it's their
Yeah.
research, it's their life. And so, and we really respect that. So I think it's a matter of, okay, we might have to wait a little bit longer for it, but we know that what comes out of it is going to be robust. And we know that even if there is a hangup or some kind of hiccup, they'll probably be able to help us work around it if we had to, because they're going to know what's best to kind of move next. And I think that and it's also not as expensive. So when you're trying to run a small business, I mean, you do obviously have to pay attention to cost, and an academic route is going to be significantly cheaper than trying to hire these big CROs or
Right.
industry partners. And it's, they're not going to have your best interest in mind. Like
Mm.
they're going to have kind of their own interest in mind. And so we really started with the academic side that for that reason. And it was, well, we knew we'd work really well together. We already know a lot of people who do a lot of fun things, and it won't cost us too much. And in some cases, it's like, I don't even know how they get away with charging as little as they do. For some people, you know, it's like, wait, you said how much? Like I would have thought 10 times that much. And so that's been great. And yeah, I know SciGen, and obviously they're similar, almost kind of like USONA or something else, trying to do more of the manufacturing and then
Yeah.
help supply to people. And that's a different beast in itself too, because I mean, obviously, so on the cannabis side in Florida, it's what they call vertically integrated. So we went all the way from putting a seed in the ground and growing the plants through process, all of the products,
Mm.
manufacturing, production, everything, all the way to we had to have our own dispensaries. And so we actually then dispensed
Wow.
all the product as well. So we were part of that entire line. And I can tell you, Chris and I, manufacturing was horrible. Like I never want to be part of that ever again in my life if I can avoid it. If I have to be, I will. I'll get it done. But it was just, you have to deal with so much more on the regulatory side, so much more on just your quality control, you know, everything, other aspects of it that I just really don't like to think about at night. And so I think that for, you know, like SciGen and USONA and some of these others, they're really taking on that side of it too. Like they're going to have to deal with
Ah.
the regulatory oversight, making sure everything is up to code, making sure their C of A's are perfect, making sure quality and all of that stuff is perfect. And that just takes a little bit of a different mindset, I think. Whereas for me, I like it to be way messier than that. Like I love the research area and then the drug
Yeah.
discovery area. And like you mentioned your box, that's sort of, my box is like that chaos zone of having to find the patterns and figure everything out, and trying to find the best path to get those things forward. So that's why we're in the process of hiring a clinical development managers, because I'll be ideal for managing people, but not necessarily running a clinical trial, because
Yeah.
that just doesn't sound fun to me. And I would love to talk to the patients and work with patients. And that aspect of it is great. When I did that in cannabis too, I loved being able to go to a patient, talk to them about the product I made, and talk to them about how it helped them. Like that was phenomenal. But if I had to be the one running the clinical trial, that doesn't sound fun. So
Mm.
we hire someone who's really good at that and let them kind of handle that part of it. And then, yeah, just help manage it. So I think it's, yeah, just finding where you enjoy yourself and also where you're good at things, and just helping people find their niche. I think the fact that you like to be in a box, that's great, because then if I ever, you know, if I need a computational person, it's like, that's the person you go for, though, is because you know that that's where they want to be and that's where they're going to do their best work. And you know that what comes out of it is going to be robust and it's going to lead to something. And I think that it's not everyone wants to be a jack of all trades. And I think that you need some people to not be that, because otherwise they're never, that's the whole statement, right? Jack of all trades, expert of none. Like
Yeah.
and you want experts sometimes too. And a lot of the time nowadays, you like the US is starting to learn to like its specialists and its experts and things. And so, yeah, you get there, but I'm not going to pretend it's easy by any means, but I think it's fun. I think that that's maybe a personality thing at that point. I don't know.
No, yeah, I
But
mean, I'm super excited about all the companies like Mind Medicine and Numinous Wellness that are just taking on the psychedelic space. It's so great to see it after being a part of, like, you're probably also a part of the Dare generation. You probably went through Dare when you were like
yes,
8 years
like
old.
elementary school.
Yeah, you
Yeah.
probably went through Dare. So
Yeah.
it's really cool to see after having been through all that stuff, I'd say.
Yeah, I used to actually, I gave this little pin. I remember it in grad school to Chris. I found it in some backpack from when I was little, or I don't know, something. And it was the just say no pin. And so I remember I brought it in and put it on Chris's desk one day. And I told him, I was like, you should keep this. You should.
That's so
I think it was
funny.
when he got the cannabis job, because he got that right out of his PhD. And so I think I gave it to him as a joke. But
That's super
yeah,
funny.
I still had that for a long time, which was funny, which I took to heart for sure when I was little. And I can see why it's hard to sort of shift people's minds away from something that's been drilled into them so hard for a long time. And I just took it a little bit more seriously when it came to like cigarettes and alcohol.
Totally.
I never really, and like hard drugs and things, like really intense stuff. But I think, I don't know. I don't know if it's just because I grew up in Florida or not, but cannabis was just like everywhere all the time. Like it was never even really like I don't know. I never associated that with the DARE program.
Mm.
To me, that was a separate thing. And maybe our DARE officers didn't focus on it as much. I don't know. But I feel like I always remember them talking way more about like cigarettes and alcohol and things. And so, and other really intense stuff. So I don't know. So that didn't take too much of my mind to really
Oh, I
change,
see.
because like
Okay.
I grew up already kind of like not really thinking that was part of it. But for someone who takes that really seriously as I hate cigarettes, I could see it being hard to then try to like steer someone in the other direction. So we have a lot of work to do. We have a lot of work just for helping people recognize the promise of these things, helping, bring education to the public, things like this, like even your videos and other things, like just really trying to help bring awareness and help people be educated and understand sort of the process. And I think there's a lot of people that are now part of that are pretty big names to try and help make it happen. So hopefully this time it sticks. But
I have one last thing that I like to ask a lot of people. Like, did you
okay.
did you have an initial spark in life that was like, I want to study chemistry? Did you have that somewhere in your childhood or in your teenage years? Or like, I like to ask this question.
Yes. So I will say there's two different major points that happened for me. And one was when I was very little. I was maybe in like first or second grade. And I remember my dad teaching me about atoms. Now, my dad didn't go to college. He's a Navy vet. He was in construction most of his life, but he was always really interested in fun, just kind of sciencey things. I think he kind of just did it almost like a hobby. And so he started teaching me about atoms one day. And I remember thinking like that was the coolest thing I had ever heard, was that like there's this thing that we're all made of, everything's made of, and it connects everything. And I was like, that makes so much sense, and I'm like
Yeah.
6, right? Or 7. Like, I don't, and so that was the first point. And I remember that vividly still. And then the second point was when I was in 8th grade and I started reading the book Alpha and Omega. I don't know
Mm.
if you've ever heard of it,
No.
but it was one of those kind of like popular physics kind of like
Mm.
books where you're learning about sort of particle decay and, just kind of astronomy sorts of things and universe stuff and all of that kind of stuff. And it was in line with the Elegant Universe came out later.
Yeah. Yeah.
That same line of types of books, right? But this one came out first, and I believe I was like 13. And I remember being in class one day. in my freshman year of high school and looking at my teacher, and he was talking about particle decay, but he like messed up something. And I remember raising my hand and being like, no, that's not right. That's like beta decay. That's not alpha decay. You got to like fix this. And I remember him being like, what? How do you know this? And I remember telling him,
Right.
and no joke, I feel like such an, like, I almost swore there. Sorry. I feel like such a terrible person. I don't know how it works.
I don't care. You can swear.
Yeah. But yeah, I felt, looking back on it, I was such a brat because I remember him telling me, like, how do you know that? And I looked at him and I'm like, I read. Like, that was my response.
I read.
I can still like remember looking back on it like, oh my God, I was so mean to this poor guy who's just having to teach all these like, you know, super crazy teenage kids. And here I am being like the smart ass in class saying these things. But anyway, that was the moment. Was I realized there that I was already learning and knowing a lot of things. And it was so interesting to me that I was like, this is what I want to do. I knew I wanted to be a scientist from like it, like that was it. I was
Wow.
sold. And so I got through like Calc 2 by the time I was 16, you know, like I
What?
was, yeah, like
Were you taking university college? Did you graduate early or something?
So my entire senior year was me basically being like a TA for teachers. Like I just didn't even, they didn't have any chemistry or math left for me in my high school because I'd
I didn't
already
even know
taken,
they taught Calc 2 in any high school. That's wild.
well, we called it
Jeez.
Calc BC. There
Oh,
was
okay,
Calc
yeah.
AB and
A, B,
BC,
and B, C,
but
yeah.
if you got a high score on the exam, it got you through Calc 2 in college.
Jeez.
So then I did like AP chemistry my junior year as well. So by the time I got to senior year, I was just like, okay, what do I do with myself now? And so I kind of just, you know, messed around and did whatever. But yeah, I knew I wanted to do science. I didn't know in what aspect. I don't know if anybody ever really does. I thought it was to go the academic route, you know, be a professor and do that sort of thing. Life kind of got in the way of that a little bit. The academic route's pretty pretentious. If you break the cycle, it's hard to get back into it. So when I ended my postdoc early to come back to help with my dad, it was just kind of like, from there I was like, okay, well, maybe there's, you know, another way for me to really get into the research and do the things I want to do. And I mean, my options were pretty much academia or being an entrepreneur. Like I knew there probably wasn't going to be another route that I was going to be satisfied with. So that's I guess why I'm here now.
That's really
But
cool. And I guess you didn't want to be Alexander Shulgin, you know,
right. And what's
working at a
funny is
lab in your house.
yeah, I mean, that would be super cool, except I do have a son. And when
Oh,
I had my son, I remember thinking to myself, like, man, that's the point where you have to start really caring like about that stuff. You can't just start doing silly things like that. So
yeah.
um, you know, had he maybe not been in the mix, I might be a little bit more adventurous with those things. But no, I definitely try not to mess around with it because of him. But what's funny is if you ever go look, like you can see him on our team page for the company, but if you ever look up my professor, Bill Baker, that we both got our PhD with, he almost looks like a like barely younger version of Shulgin. And
That's
so it's
funny.
just so funny. Like I didn't even really connect it until just this past year. All of a sudden, I was looking at the back of TCal or like wherever his image is on it. And I remember being like, oh my God, how did I never really make that connection that he looks so much like him? But yeah, just kind of crazy, wild, you know,
Oh,
white hair
yeah.
and beard and stuff. So he's always wearing Hawaiian shirts. It's like
Yeah.
this, and he wears Birkenstocks everywhere. But yeah, so it's, who knows, maybe he was a huge influence on sort of my career paths and things too. So yeah.
Yeah, well, in keeping in with the time, I don't want to use any of your time. I really thank you so much for talking to me.